My name is Adede. I am your co-moderator today together with Gloria, who will also be speaking a bit later on. We have an amazing lineup of speakers today, and there's a lot of information that's gonna come our way. So let's be ready to listen out. I'll introduce our first speaker, and I can see he's already in the meeting. So as he gets ready, I'll just introduce Dr. Shakir Atoyebi. He'll be speaking about the ethics that surround the use of AI and digital tools. So Dr. Shakir holds a Bachelor's of Pharmacy and Master of Science degrees from Nigeria. He completed his doctoral studies at the University of Liverpool with the Duncan Norman Research Fellowship, and he is currently a postdoc research associate at the Institute of Systems, Molecular and Integrative Biology at the University of Liverpool, where he leads the modeling component within the perinatal pharmacology group. So we are excited to welcome Dr. Shakir. Please, let's give him a round of applause through our emojis and reactions. Let's welcome him as he shares his screen. And please feel free to use the chat to type in all your questions. We may not have all the time to have everyone, speak as and ask questions, but let's use the chat. The speakers will be ready to answer your questions in the chat. So don't be shy about typing or even raising your hand to unmute. So Dr. Shakir, you're welcome.
Hello, good evening.
Hello.
Can you hear me clearly?
Yes, you're clear.
All right. All right. Hi, everyone. Good evening and good morning. I would like to thank the organizers for the honor of the invitation to present at the anniversary. So today I'll be talking about bridging the gaps between research ethics and digital tools in the 21st century. My name is Shakir Atoyebi. So, yeah. So a little disclosure. Uh, it's a bit stuck. All right. So a little bit disclosure, and that's some large language models were extensively used in developing some of the slides that I'll be showing. So I'd like to acknowledge them, but not as authors. And I also appreciate in advance that my presentation is quite text heavy. So please pardon me. Now, this is the outline I'll be following today. So I'll talk a little bit about ethics and research, then we'll move on to artificial intelligence. Then we'll dive into some of the common challenges with AI. We'll look at some of the recommendations, how we could bridge the gap and some opportunities and conclusion. Then there's also some materials for more reading. I'm happy to share the slides at the end of the presentation. So to begin with ethics and research. So when we talk about ethics, the Oxford Dictionary defines it as moral principles that control or influences a person's behavior. It could also be described as a system of moral principles or rules of behavior. Now, if we bring it that to research ethics, now this kind of refers to the moral principles of planning, conduct, and reporting of research. It encompasses protections for human and animal subjects in research. But you would also agree with me that there are also many, even there are many researchers, even those in the bioscience field, that conduct their research, but they don't deal with live human and animal subjects. So it has broader ethical challenges. It encompasses the collection, use, and interpretation of data, methods for reporting and reviewing findings. And it also covers relationships between researchers and how we respond to misconduct. Now, the purpose of research ethics basically is to foster research in a manner that it protects the interests of the public, the research subjects, and researchers themselves. I would now move on to talking about a very famous case of research misconduct, and this is the Tuskegee syphilis study, which was conducted between 1932 and 1972. Now, this was conducted by the U.S. Public Health Service in collaboration with the Tuskegee Institute. Please, can I confirm you can still hear me?
Yes, yes, we are all following.
All right, thank you very much. So the aim of, the nefarious aim of this study was to observe the natural progression of untreated syphilis in African men. And the researchers falsely claimed that they were treating the participants for bad blood. Now, approximately 600 black men from Alabama were recruited for this study. About 399 had syphilis, and 201 without syphilis were enrolled. Now, there were some, they were improvised. These were people that were very impoverished, and they were promised free medical exams, meals, and burial insurance. Initially, the study was planned to run for 6 to 9 months, but sadly, most sadly, the study continued for 40 years, and not until it was exposed by the press in 1972. Now, it is very obvious that in this study, there was, lots of ethical violations, which included no informed consent. The participants were misled and denied effective treatment when it became available. And the standard of treatment in the mid 40s was penicillin to treat syphilis. So even though the there was treatment for the ailments, for the disease, the participants of the study were not offered this treatment. They were misled and denied effective treatment. Rather, they were given placebos and ineffective procedures. So part of the consequences of this was at least 28 men died directly from syphilis. There were about 100 from related complications. About 40 wives, spouses of the men recruited for this study were infected, and 19 children were born with congenital syphilis. Now, as you can imagine, this led to a huge scandal. This scandal, which also resulted into a class action suit. There was about 10 million US dollars settlement, and it also led to major reforms in the US research ethics. And ultimately, you had the National Research Act and the Belmont Report. So this research misconduct, it kind of underlines how bad, how wrong, how much things can go wrong when we don't have systems in place to actually checkmate, whatever uh thoughts people have about it'd be nice to test this, it'd be nice to test that. So there is need to have some moral principles that protect both the research subjects, the researchers themselves, and the collective society. So there are some other instances of some research misconduct, like the Nazi medical experimentation in the 1930s and 1940s. And you also had the research conducted at Willowbrook State School in the 1950s and 1960s. So you can, under the See More resources at the end of this presentation, you can, once I share that, you can read more about what pertains to this misconduct. So from looking at the case of the research misconduct, it's very clear. The importance of research ethics. We need it to protect research participants. We need it to ensure that there's credibility in research and reproducibility of findings. We also need it to ensure that to promote global collaboration and trust. And it also helps us to uphold integrity in biological research. Now, I'll be changing gears a bit and moving on into artificial intelligence in biological research. So when we say artificial intelligence, AI, it can be defined as that it refers to technologies or tools that can perform human tasks which require intelligence. That is, any human task, tools that can perform human tasks which require which requires intelligence. Now, what kind of intelligence are we talking about? Intelligence such as perception, judgment, reasoning, or decision making. Now, we've had lots of computer programs, cell phone apps, electronic calculators. So are these AI? No, we wouldn't call them AI because they are not particularly associated with intelligence because they do not learn from the data. Now, machine learning is a branch of AI which focuses on using the data and algorithms that to enable artificial intelligence to imitate the way that humans learn. And it also works towards gradually improving its own accuracy. So it kind of learns. It's quite a bit complicated, but I would definitely recommend this article for us. I mean, for us to go to read. I've tried to see if I can find the. Okay, I'll just move. I'm trying to see. All right, so I would definitely recommend this article for us, and I've attached uh like a link to it in the documents. So for those who want to do more reading. Now I'll move on to generative AI. Now, this refers to deep learning models that can generate high quality text, images, and other content based on the data that they were trained on. Now, large language models such as chatbots like ChatGPT, Gemini, Copilot, and the likes, they are large language models, and they are also a form of generative AI. Now, these are able to analyze, paraphrase, translate, and generate different contents, which could be text, which could be images, and other forms of content. But how do large language models, how do they work? Now, they are statistical algorithms that are trained on huge sets of natural language data, such as text from the internet, journal articles, books, and other materials. Now, by processing these data, these large language models they learn to estimate probabilities associated with possible responses related to text, and they can rank responses according to the probability that they will be judged to be correct by human beings. So if you say something, I mean, if you put a prompt, they are going to look through the internet and try to see, oh, where have this maybe this set of words, where have they occurred? And which kind of they might look at all the sources, I mean, all the available resources that maybe those keywords have appeared, and they kind of try to rank them and try and see which one might be most relevant to the question asked and things like that. So some uses of AI in research. Uh, AI has been, I mean, it's often used in analyzing and interpreting data and images, uh, generating hypotheses, modeling complex phenomena. And I think generally most of us would you might have used AI for searching and reviewing scientific literature, for writing and editing, uh, papers, grant proposals, consent forms, and many other uh, forms of writing, and in to review scientific papers and other research outputs. Now, some uses that are more related to biological research, have been used, I believe, in genomics and proteomics research, bioinformatic platforms, and a few other uses. So, what are the common challenges with AI? Now, AI is not error-free. So sometimes they could give you results that diverges away from the standard of correctness. So you could have random errors. And this, I mean, quite a lot of people have reported citation errors. Like it seems like, the large language model is hallucinating. Like it's saying something that you could, maybe you feed it with a paper or you ask it a question and it's saying something that, or it cites a paper that doesn't exist It's something I've actually personally experienced myself before, and it was shocking. So, similarly, you could have biases in AI algorithms. Now, AI algorithms are I mean, they are designed to represent the data that has been used to train them. So any bias in those data, would also feed into feed into their performance. So they can reproduce or amplify racial, ethnic, gender, and other forms of biases in the training data and data received later. There has been reported racial and ethnic bias, which impact the use of AI in medical imaging, diagnosis, and prognosis. You have more details or references in this paper cited below. So, I mean, just one thing to mention here is in recent time, there's been a lot of clamor about the fact that you have I mean, a lot of data are actually available in biological research are data gotten from male subjects, and not even only humans now, but even animals. Oftentimes, many researchers, in many cases, it has been shown that a lot of data they would often kind of focus more on generating data on using males than female subjects. So you can imagine whatever biases you have with regards whatever gender bias you have in the data that is available, if that is used to train the AI, that could also be reproduced when it is being used later in the future. Now, another thing to also think about is the fact that AI it lacks moral agency. There's been multiple instances of AI, people using chatbots, directing them to harmful practices. A number of people have implicated that AI has uh, kind of guided or even supported their loved one committing suicide. There has been a case of a 10-year-old girl that was when she asked Alexa for a challenge, she was asked to stick a penny into an electric outlet. So sometimes, I mean, it can't really say, oh, this is good, this is bad. It's just whatever you want, whatever you feed into it, it tries to support you. And similarly, you also have the black box problem. That is, it is very, very difficult to really, nitpick and, to dissect how it works, how it has been trained. And you can look at, the whole process, but the nitty-bitty of the things, how it's as ascribed weightings to really improve itself, it's very, very difficult to try it because there is just thousands and thousands of information and notes to follow through. Now, this challenge also translates to ethical challenges because, as you can imagine, errors would undermine the validity and reliability of scientific knowledge, which could be disastrous and which could offer disastrous consequences for public health and safety. Bias could also undermine the quality and trustworthiness of science, and it could have social impact. Now, we also have challenges with data privacy, ownership, confidentiality. Now, for most parts, when you use large language models like ChatGPT, Copilot, and a couple of others, they store the data that has been submitted by their users. And I think some of it has been said that some of them might have the opt-out option, but if you don't effect the opt-out option, then whatever input or data you have put in could be used in further training the AI and things like that. And that's one of the reasons why it's always very advised that not to include personal or confidential information, not to feed personal or confidential information into these tools. Now, there has also been concerns about misuse of sensitive biological data and quite a host of other problems. Now, some recommendations has been offered by some researchers, and that includes that researchers should be held responsible to identify, describe, reduce, and control biases and random errors from AI. It's important to also disclose, describe, and explain their use of AI in research and limitations. It's also important to engage communities that might be impacted on issues related to bias. And where researchers use synthetic data, it's important to indicate which parts of the data are synthetic. It's important to label the synthetic data and to describe how the data was generated in the first place, and also explain how and why the data was used. Now, it's also recommended that AI systems should not be named as authors, uh, inventors, copyright holders. However, their contributions to research should be disclosed because, I mean, you cannot, I mean, in a way, I mean, they cannot be held responsible uh, for, uh, it's difficult to hold them to account in a way. So it's difficult to say, oh, they should be listed as authors or inventors and things like that. Now, it's also important that there should be a lot of education and mentoring with regards to responsible conduct of research. And this should include discussion about ethical use of AI. And I mean, just like any other tool, artificial intelligence is something that can be used for good and negatively. So it's now important that whoever wields the sword wields it well. Now, how can we bridge the gap? So there is a need to integrate research ethics with modern technologies. It's important for us to strengthen ethics review processes, particularly uh, as might be affected by AI. It's necessary that the younger generation and everyone try to build literacy and digital skills in AI. It's important for us to also collaborate with global research networks towards achieving this and advocate for clearer policies and guidelines. Now, what are the opportunities out there for young African researchers? I mean, it's to a large to an extent, AI it kind of levels the field a bit. So it would be nice if we are able to leverage open source AI tools for research. But it's important that as we do this, that we participate in ethics and AI training programs. We should engage in interdisciplinary projects and try to contribute to developing Africa-centered ethical policies because we are the future. Now, in conclusion, ethical integration with AI tools is vital for credible research as time passes. Now, young African researchers can lead in responsible innovation. Collaboration, education, and advocacy would be key to bridging these gaps. Thank you very much. So
Thank
these
you
are
so
some of the resources I have hopefully, when I share the slides, you can go through. Thank you very much.
Thank you so much for just taking us through everything from the very beginning of issues that were there with ethics before AI, and then coming in to just speak about now that AI is here. It's a tool that's here to stay, whether we like it or not. And it's something that we need to embrace, and we need to be very ready to, like look at the pros and cons of it and see how best we can learn, how best you can adapt. So thank you so much for that. We still have some time for questions. So if there's anyone who would like to ask anything um, about the presentation that we've had. So I'm just looking at the chat. I can see some comments coming in. You're also free to raise your hand. Nasiruddin says, thank you for the brilliant and insightful session. Martin also says, thank you so much for the presentation.
Thank you all. You are very kind.
When Yeah, I also say thank you so much, and also thank you for just expounding on the Tuskegee study. I remember when I first learned about it, I was so shocked, and every time anyone repeats the whole story, I'm still as shocked. Like I never get less shocked. So just that just shows what can happen when ethics are taken away from, the research that's being done. And I think with the advent of AI, I'm sure everyone here has interacted with some AI content that you deem questionable. So I don't know if it's just me on my end, but if you have had any interactions with the AI content that should be like, okay, is this really right? Because I know some people can use it and maybe uh bring in issues like cyberbullying, because it's something that was made by AI. So trying to trace back to whoever did like what exactly, and then now in our world of biosciences, there's also a lot that can be done. So thank you so much. Let me just see if there's anyone who has a question. Emmanuel also says thank you so much for the presentation, and I can see many reactions. Dolapo, Taufikat, thank you so much everyone for your interactions. And remember, Shakir is still around, so you can still type your questions in the chat, and he will respond even as the other presentations are ongoing. So we'll move forward to our next presentation. And the presentation we have next is a poster presentation by Paul Owolabi. So the poster showcases some collaborative work that he has done at the Department of Neurosciences at West Virginia University, and it's titled Chronotherapeutic Administration Alters CAR T-cell Trafficking in HER2-positive Breast Cancer Brain Metastasis. So Paul, if you can hear me, oh yeah, I can see you here. So kindly just share your poster. You'll have 5 minutes to take us through the poster, and we'll also have 5 minutes of questions and answer.
Hello, can you hear me?
Yes, you can.
Okay. And could you please confirm that you can see my screen as well?
Yes, it's clear.
Okay, thank you very much. First, I'd like to appreciate the Biospherea community for this opportunity, and I also want to say congratulations on the fifth anniversary. So, today I'll be presenting my poster titled Chronotherapeutic Administration Alters Capital Trafficking in HER2 Positive Breast Cancer Brain Metastasis. Sorry, 1 minute, please. Okay, so talking about breast cancer brain metastasis in general, metastatic breast cancer remains one of the leading causes of cancer deaths in women globally, and brain metastasis in particular is also very life-threatening, and it occurs in about 30% of patients with metastatic breast cancer, particularly those that overexpress HER2, which is the human epidermal growth factor receptor 2. And due to this huge, significant health challenge, there is the need to develop effective treatment strategies to mitigate these challenges. And one of these treatment strategy is the use of CAR T-cell therapy, which is a cell-based therapy which involves engineering T cells in the body to convey specific antigens known as the chimeric antigen receptor, which specifically targets antigens in the cancer cells and tries to eliminate them. And although this CAR T-cell therapy has been very effective in the treatment of hematological cancers like lymphoma, myeloma, and other forms of liquid tumors, they've been very challenging uh, in the treatment of solid tumors like breast cancer, pancreatic cancer, and other forms of solid tumor. However, recent studies have demonstrated that by altering the time of day of administration of CAR T-cell therapy, you can improve their efficacy and like the overall survival of the patient. And so, because of that, we wanted to see the effect of time of day in the administration of CAR T-cell therapy in breast cancer brain metastasis. How it's wanted to test the trafficking as well as infiltration based on administering at different times of the day. And to do that, we injected mice, NCG mice specifically, female NCG mice, which are triple immunodeficient mice, meaning that those mice, they lack functional NK cells, T cells, and B cells. We injected them with GMT1 BR3 cell, which is a HER2 positive breast cancer cell that have been enriched for brain tropism. So these cells, when we inject them in the left ventricles of the mice, they migrate to the brain and form metastasis there. And after a period of 21 days, we inject the CAR T cells through tailvein injection and then collect the brains uh, 24 hours later. One thing I want to point out is that we injected at two different times of the day, which is at ZT1 and ZT13. This ZT is a circadian nomenclature, which means ziggurat time, meaning that ZT1 is 1 hour after lights on, and ZT13 is 1 hour after lights are off, which represents the inactive and the active phases of the mice. And noting that mice are nocturnal animals, most mice used in laboratory are nocturnal. They are active at night and inactive during the day. So next, the figure here just shows process in which we use in generating our CAR T cells, which involves activating those T cells that we isolate from PBMCs from patients. We activate them with IL-7 and IL-15 cytokines, and then we transduce this with a lentiviral vector that carries the chimeric antigen receptor. And a chimeric antigen receptor construct has an anti-HER2 signature invariable fragment connected to CD28 transmembrane domain, and as well as the CD3 zeta cytotoxic domain. So now moving to the results, what we observed was that we observed because of time, we observed more trafficking at ZT1 compared to ZT13. So what we did was that when we collected the brains, we processed them using cubic brain clearing. We did light sheet microscopy to image them at different magnifications, and then we quantified the CAR T cells that were able to infiltrate into the tumor site. And from all of these figures, we saw more uh, CAR T cells trafficking at DT1 compared to DT13, as is shown here. I will show that it's not specific to a particular mice across different mice. And then we also showed here, we showed more infiltration. So in this case, in the case of figure D, it's measuring how close the CARs are to the tumor. So when we measure the tumor and the CAR, we try to determine the shortest distance between them. The shorter the distance, the more closer the CAR T cells are to the tumor, which shows that there's a huge amount of infiltration within them. And the figure here is showing it's just a visual representation of what the data that we have in figure four. In figure four talks about. Then the next thing that we did was that we tried to see if there are region differences between this CAR T cell infiltration and trafficking. So we quantified according to different regions of the brain, the anterior region of the brain, the middle region of the brain, and the posterior region of the brain. And across these regions, there is the same effect was observed both in terms of trafficking and in terms of infiltration as well. And the next step that we did was to determine the functional relevance of what we have studied. So we did a co-culture of the CAR T cells with our cancer cells in vitro to determine the functional relevance. So in this cytotoxic assay, we across different effector to target ratios, meaning that the ratio of our CARs so the ratio of the tumors, we can see increasing cell death across this effector to target ratio as compared to our baseline, which is the non-transduced T cells, which are just normal T cells. And so this shows to us that there's functional significance of the CAR T cells being able to kill the tumor cells. So in conclusion, the mice that received the CAR T cell injection at CT1 showed over 90% increase in the percentage of tumor-associated CARs, as well as infiltration within breast cancer brain metastasis relative to ZT13. And these results suggest potential chemotherapeutic benefits of intravenously administering HER2-specific CAR T cells against um, HER2-positive breast cancer brain metastasis. So in terms of future directions, the next thing we're going to be doing is to run a survival study, that is inject the mice at those 2 time points and allow them to go on for a long period of time and assess the overall survival of those mice based on those treatments. And the next set of experiments also will be to try to conduct mechanistic studies to uncover the mechanisms that are driving the time of day dependent trafficking and infiltration. So at this point, I would like to thank the Biospherea once again for the opportunity, and then wait for questions or comments. Thank you.
Thank you so much, Paul, for sharing. I think that's very dense topic to speak about in such a short time. I think you covered all the major sections of it, and especially at a time when there's a lot of research into therapeutic approaches that are best suited for different types of cancer. So thank you so much. Yes, I see one question which we will take because we have a minute to go. Titilayo is asking, does this work for breast cancer cells that have metastasized into the brain? So, Paul, could you take up that question? Does this work for breast cancer cells that have metastasized into the brain?
Um, I would say yes, but this is still in the preclinical stage, so I wouldn't say definitely. So this study is mostly in terms of showing that by just altering the time of day, we can increase the amount of T cells within the brain. So just altering the time of day, we can increase the amount of T cells. So whether those T cells are effectively able to do the killing, it remains to be seen. So it depends on further study showing that that those T cells being available within the brain in large quantity and in significant amounts are able to do the killing. So future studies would show that. But yeah, hopefully it's able to eliminate the brain metastasis within the brain. Because in terms of CAR T-cell therapy, some of the reasons why it's not been effective with solid tumors because of poor trafficking. For instance, in the brain, we have the blood-brain barrier that limits trafficking to the brain, and we also have other challenges with the toxicity level as well as the immunosuppressive tumor microenvironment. So this is just kind of addressing one of the challenges.
Okay, thank you, Paul. I I hope Titilayo is satisfied with that answer. I think that was a good and comprehensive response. If anyone else has any other question, kindly type. Okay, yeah, Titilayo says, all right, and thank you. So if anyone else has any other question for Paul, you can type it in the chat. I can see Shakir responded to the other questions. So the chat is effective. Then we'll just move on to the next presenter, who is Mr. Raphael Aminu. So he's going to speak to us about AI and the future of work. So just a brief introduction as Mr. Raphael gets ready to share his screen and speak to us. He is the founder of Bioscite, and he's also a research consultant dedicated to study of malaria and trypanosomiasis. He's big on connecting academic research with real-world applications, and is very focused on improving health outcomes on a global scale. So his research contributions have been recognized at the Africa Bioimaging Consortium Travel Award, and also the Sao Paulo Research Foundation grant. So we are looking forward to listening and engaging with Mr. Raphael Aminu. I don't know if Mr. Raphael can hear me and share his screen. So,
Hello, hi everyone. Good evening. My name is Raphael Aminu, and I welcome you to this presentation and the celebration of 5th anniversary of Biospherea AI. My work is made a lot easier by the early presentation by Shakir. So I'll be discussing on AI and the future of work, positioning African young bioscientists. However, I'm a PhD student at the Department of Biochemistry at Adegboyega University's area. So to begin, this is a brief outline. My aim this evening is to stimulate our interest on how to embrace AI for future research and how to position ourselves for better global opportunity. AI, as earlier mentioned, is artificial intelligence and the use of so many subsets of AI such as big data, machine learning, and automation. Bioscience has undergone a revolution driven by AI, and the time to embrace AI is now. There is no any other time. Just 2 days ago, I was discussing with a few of my colleagues, and a lot were said that AI will make people lazy, but that's not true. AI has come to stay, and AI has come to change how we think and how we process data in life sciences. Africa, the imperative. Africa has the world's largest, youngest population, and hence the need to empower this population with the critical tool needed to succeed as bioscientists. And this will be so important in solving African unique health challenges, agricultural challenges, and well as climate issues. Nevertheless, AI has undergone some transformative role, especially in areas of drug discovery, where AI uses algorithm to analyze big data, big molecular data to identify drug targets, reducing R&D time cycle for years. Interestingly, during COVID, it took much shorter time to find a vaccine for COVID-19. And recently, time required for drug discovery is meant in enhanced or rather reduced by the use of AI and machine learning. There are also areas of precision genomics where big data or machine learning models are used to analyze different variants of personalized medical insights tailored to specific genetic profiles. This is made possible only by embracing AI and hence the new order. Also in agriculture, AI has been used to solve and also dictate through early detection of diseases among crops, which is also critical because in Africa, food security is also a very important issue. Strategy. In an attempt to encourage African young scientists to embrace AI, there is need for young scientists to speak two languages, biology and code. We need to change at the fundamental of our curriculum, a shift from mandatory coding, integrating Python and R programming into undergraduate studies for biology. However, I think at this point, some of us have started using these mandatory coding languages as postgraduate students, not as undergraduate. But now, this platform has created an opportunity for us to understand that there is need for us to understand biology and as well as coding language. It's not only for the computer guys. And also they need to also focus on hackathons, local sessions that will help us to look at disease such as malaria, trypanosomiasis, among others. Digital literacy also is very important through training of cloud computing and bioinformatics pipeline. However, I think in Nigeria, there are a couple of good organizations that have engaged in digital literacy. I think Helix Biogen Consult is one of them, and I think they've done extensively well in encouraging and attracting young scientists to their training and internship programs. Positioning for global opportunities. To position for global opportunities, there is need to develop relevant skills and knowledge. As earlier mentioned, the skills for programming, for coding is very relevant for young African scientists if we want to position ourselves for global opportunity. Network and collaborating is very important to network and collaborate. One of the opportunities and platform for networking and collaborating is this event organized by Biospherea to mark their 5th anniversary. So it's important we take advantage of these interactive sessions and this event as we close today. They need to stay updated and on industrial trends and breakthrough. It's very important that the young African bioscientists should also be up to date on the movements and happenings within and outside industry, both in research and academics, and embrace a lifelong learning. Also, for African young scientists to enjoy this, there is need to reduce reliance on foreign clouds alone. There is need to develop high performance computing for African bees that is localized and centered in Africa. And there's also need to change from sample export to knowledge export. This is critical in terms of where there is knowledge sharing that would encourage young African scientists to embrace AI and integrate AI in learning and as well as in research. The future of global bioscience is intertwined with empowerment of African young scientists with African scientific talent. This is critical because of our population and immense talent that Africa is blessed with. In conclusion, African young scientists can thrive in AI-driven future of work by acquiring relevant skills, embracing innovation, and positioning themselves for global opportunity. A call to action. They need to take the first step, explore online courses, attend workshops like this, join professional network to enhance skills and career prospect. Thank you for listening.
Thank you so much, Raphael, for this insightful session. And we really enjoyed this. And then it's the call to action is for every one of us. Let's get our hands dirty in 2026 and learn the codes, the languages that we need to learn to be able to position ourselves rightly, right, for global opportunities with the use of AI in bioscience. Thank you once again, Raphael, for this wonderful presentation. I don't know whether we have questions. Anyone that has question, we will be able to take just one question. Why any other person with another question should drop in the chat box. Do we have any question? Yes, there is comments in the chat box from well, it says, thank you so much, Raphael. Emmanuel says, thank you so much for sharing. So let's get our end 30 in 2026 in our research interest. Let's take up opportunities to learn. Let's take courses. Let's attend workshop, like I said, and let's network and then get those skills so that we can position ourselves rightly. Nasiruddin says it was quite inspiring and informative. Muhammad said, thank you, Raphael, for all you do. All right, there is a question here from Emmanuel. That is the only question we'll be taking for this session before we move on to the next one. And the question says, what are specific courses that can be learned in relation to AI? Right, this question is for you, Raphael. You can take it now.
Thank you for this question. I think the presenter before me, the two presenters before me, I think Dr. Shakir did a great job by saying there are a lot of courses that can be learned on AI. And you can start with R programming courses, coding courses as well. These are for the basics. Sometimes we find this quite boring and time consuming, but they are here to stay. We must embrace machine learning. I think so. For us to adapt, we need to adapt to these new courses, and we need to start from the basics. There are no shortcuts. So we can start from the basic programming languages, basic coding, and then we take up the steps like that.
All right, thank you for the response. Yeah, Adegboyega Babatunde mentioned that you can start with prompt engineering, AI automation, and machine learning. That's the response for you, Emmanuel, in the chat box. All right, so we'll be moving next to our poster presentation. We had one before. There is another one that Babatunde Adegboyega will be taking. Adegboyega will be taking us on the poster presentation, molecular modulation of apoptosis and inflammatory genes by nymphaeolos in diabetes-induced cardiac injury. And it's from the Department of Biosciences and Biotechnology, University of Medical Sciences, Ondo City. All right, you, Babatunde, you can share your screen and then take us through the presentation. You have 5 minutes. You can start now. Thank you. Babatunde, we cannot see your screen yet. Is there something that we can do for you to make it?
Good evening, everyone. I'm about to share my screen now.
All right, thank you. Okay, why he shares his screen, I would like every one of us to drop in the chat box maybe a code or a language that you wish to learn in the coming year to better position you, right? You can just drop them in the chat box. If there is a course you want to take concerning coding or any language you would like to learn, you can drop it in the chat box. All right, let's keep it coming. And let's drop them in the chat box while we wait for Adegboyega to share his screen. Jenny say Python. Shakir, did you say Julia? I've not heard about that before. Adede said her. Abdulafi said, is there any code that can print modeling? I don't know. Maybe you can tell us when you find one.
Oh, yeah.
Taufika said R. Oluwashemilogo says R and Python. Adegboyega, are you having any difficulty in sharing your screen?
Yeah, my MacBook is asking for privacy and security. Just 1 minute, please. I'll share my screen now.
All right. All right. Florence says R and Linux. Yeah. I would love to learn how to. Maybe let's see what next year is. All right. So you can keep it coming. You can keep it coming. We are more than this, right? Just drop them. Just drop them. There is any for people that have not heard about Julia before, Shakir says it's a dynamic general purpose programming language. And the distinct parts include a type system with parametric polymorphism. Wow. The use of multiple dispatch has a core programming paradigm, just-in-time compilation, and a parallel garbage collection implementation. Whoa. Wow. That's I'll just add that to my archive. Maybe one day I would explore it. Nasiruddin says R and Python. Learn more, yes. That is why we are having these sessions. Right. Abdullahi says, Julia is a book people. Ooh. Okay. All right. Thank you. Okay. Okay. You can keep it coming. Keep it coming. There are some people that have not dropped, or you don't want to position yourself better next year. I would love to. Olamide says she looks forward to learning about the ethical use of AI in research, the one that Dr. Shakir talked about, right? Yeah, Olamide, learn more, right? Thank you. I think yes, Adegboyega's presentation, poster presentation is up now. Adegboyega, if you are ready, you can unmute and then speak. Yes, Abdul Bashit, the sessions, both yesterday's and today's sessions were recorded and they will be made available on our YouTube page, Biospherea Africa. You can just check them out after we are done. Right. You can go ahead, Adegboyega. Yes, we can see your screen clearly.
Good evening, everyone. I'm sorry for the delay. Thanks for having me, and congratulations to Biosphere for the fifth anniversary that we are celebrating. My name is Benjamin, and I'll be presenting my work on a poster presentation of my work on molecular modulation of apoptosis and inflammatory genes in diabetes-induced cardiac injury. The diabetes mellitus is among the top leading cause of human mortality worldwide, and patients with the diabetes are at risk of developing different heart diseases, e.g. coronary artery diseases, heart failure, and all, and eventually they succumb to these heart diseases. One of these complications of the diabetes is diabetes cardiomyopathy, and is a severe diabetes related condition where metabolic disturbance damage the heart tissues. It progress from early diastolic dysfunction of heart, and is driven independently insulin resistance, high insulin level, and high glucose level. So this my study is aimed to investigate the molecular modulation through which nephrologists, also known as Egyptian lily, can provide its cardioprotective effect in diabetes induced rats. My research question: can this plant Nephilotus also uh, ameliorate the diabetes metabolic disturbance in the heart, and also can it also um i- i- can it also reduce the effect of the diabetes on the heart? So my material method, a repository rat samples were used because I work on heart samples. So I um I did a gene expression analysis to to see how genes that are involved in inflammatory apoptosis and oxidative stress. So I designed a primer using NCBI, and I also I did computational analysis on the primer to check if they can bind to my specific gene. So I did RNA extraction on the art samples, and also qPCR. I quantify my RNA, then I use R programming language to analyze my CCT value for my PCR results. My results, all this graph was generated using R programming language for my data. So each of the graphs show the level of expression of each gene. So each of them show the level of expression of each gene, and also we use metformin as the standard drug to compare if the phytochemicals from the plant can actually perform better than the FDA approved drug. So, and also the plants we use those dependent dosage for the rat at 400 milligram at 150. My results. So, from this graph, this all this blue square space shows how the plant actually e- exerts its cardio protective effect on the heart, on the cardiac heart injury, and also the based on those dependent of the plant, 50 milligram are actually perform better than metformin. For 400 milligrams, so shows signs of toxicity on the heart. So 50 milligram perform better, and also it reduced the expression of inflammatory gene, e.g. interleukin alpha and TNF alpha, and also it also downregulates it upregulates the gene expression of apoptosis gene, and also it reduces the oxidative stress in the system. Can you scroll down, please? No, no, or the implication of the study. I mean the implication of the study. So this study actually demonstrate the potential of the plants, and it can serve as a therapeutic agent to reduce or to mitigate the diabetes induced cardiac damage, and 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 also it can serve as like a future cure for cardiomyopathy, because um there is no treatment for it now, even though people can use metformin to maintain the diabetes, but there is no particular drug that they use to maintain the diabetes complications. So the plant can serve as a future drug for cardiomyopathy, and it also provides the fact that natural products can be used in managing the diabetes cardiomyopathy. My conclusion, Nifelotus effectively modulates the apoptosis inflammatory and oxidative stress and their pathways, and it also suggests as a cardioprotective agent against diabetes-induced heart injury. These are my references. Thank you for listening.
Thank you. Thank you so much, Adegboyega, for this wonderful presentation. Thank you for showing us the potential of natural plants in mitigating diabetes-induced cardiac injury. All right, thank you so much. If you have any question would like to take one question. If you have any question for Adegboyega, you can raise up your hand and unmute to ask, and every other person can write their question in the chat box while you attend to them. All right, is there any question for Adegboyega? Any question at all? Thank you so much for this presentation. Thank you. There are some comments for you, Adegboyega, in the chat box. Raphael says it's a very interesting research. Yeah. Wealth says thank you, Adegboyega. Ruth says well done. Ibrahim says thank you, Babatunde. Nice presentation. Itilaya says well done. Well done. Nasiruddin says that was cool and an ambitious research. Well done, Adegboyega. Thank you for the kind words. Blessing says, thank you for sharing your research with us. Right. There is a question from Wealth to you, Adegboyega, and it says, what kind of toxicity did you notice at high concentrations? You mentioned that at 400 milligram, the plant shows signs of toxicity. Wealth is asking, what kind of toxicity did you notice at this high concentration? Adegboyega, can you hear me?
Yes, I can hear you.
All right, can answer the question.
So thank you, Okete, for the question. So the gene was overexpressed with 400 milligram, both for the inflammatory gene and also for the apoptotic gene for 400 milligram actually show for pattern of RNA damage, because I use R to compute my result, so it shows a sign of RNA damage. So, but for the for the inflammatory samples, it has high inflammatory gene expression.
All right. Thank you, Adegboyega, for the response. If you have any other question, okay, there's a question from Ridwan. Adegboyega, you can attend to that in the chat box. While we move on to our next speaker, Dr. Kolapo Oyebola, who will be taking us on the topic from lab to leadership, building a high impact career in bioscience. So I'll be taking a brief introduction of Dr. Kolapo Oyebola. Dr. Kolapo Oyebola is an expert in genomics, bioinformatics, data science, and machine learning with advanced proficiency in Python, SQL, and Linux, and hands-on experience deploying cloud-based solutions. He is the director of Center for Genomics Research in Biomedicine at Mountain Top University, Nigeria, and also the founder of Business Development Bank of Canada, founded Telecare Medical Rears. So we have the honor of having Dr. Kolapo in our midst, and then he would be taking the next presentation. Thank you, Dr. Kolapo. You can unmute and speak and also share your screen. Let's make some noise for Dr. Kolapo while he takes his presentation. I want to see the excitement in us while he starts. Let's drop some fire emojis so that he can see that we are ready for him.
Thank you. Thank you for
All right,
having me.
Dr. Kolapo.
Yeah, thank you so much for having me. I really appreciate and commend the organizers of this program. Um, I'm excited because it's indigenous, and I want to believe that the sky is the limit, and there will be a lot of great things to come out of this now and in the future. So today I've been told I have 30 minutes, so I just have to stick to time. Share my entire screen. Okay, share my entire screen. So, yes, I'll be talking about how to uh, navigate, from being a lab assistant or lab researcher to a scientist to being a leader, the research leader, and how one can build a high impact career in bioscience. Caveat, not like I'm there, you know, I'm not on top of the pyramid, so I'm also climbing. So whatever I say here also applies to me. Okay. Um, and another caveat is that no two career pathways are the same, right? So on that note, I will not be sharing my own personal experience because it's never possible for two career pathways to be exactly the same. Your pathway will be unique and it's never linear, right? So it will come with a lot of bends, a lot of give and take, and then of course, it's never the same with the next person. So I'm not going to be sharing personal experiences here. So my name is Kolapo, just like I've been introduced, and I'm excited to share this with you. So, what's the bioscience landscape like, right? If you have chosen a career in science, what do you expect to do, in achieve, at the end? What kind of career do you expect to build, by choosing a field in science, especially bioscience? So yes, you can be a researcher in the university or in a research institute. We have so many research institutes in Nigeria, for instance. I don't know how widespread the audience is, but of course, if you want to think about the biomedical research institute in Nigeria, you'll be thinking about the Nigerian Institute of Medical Research in Lagos. You'll be thinking about IITA in Ibadan and so many other. And of course, you have research institutions across different colleges of medicine, across all the states in Nigeria. So you can be a researcher in the university. Unfortunately, that is not well developed. The pathway of being just a research professor, um, a research academic in a university is not well established. So you tend to focus more on being a teacher more than being a researcher in the university in Nigeria. But that pathway exists. You could just be a researcher in the university. And you could also work as a research scientist in the university or in the pharmaceutical industry or in the hospital, right? This comes after you must have achieved or attained your MSc or PhD, right? You could be a lab scientist in the hospital. You could be a toxicologist. I think someone mentioned that in the chats sometime ago. And then, of course, you could be a pharmacologist, laboratory analyst. And of course, you could just be a teacher in the university, like a lecturer, and then advance to be a professor. Um, but things are changing, and the landscape is also changing with the advent of bioinformatics, data science, machine learning, AI. You could you know be a data analyst, a data scientist, or a machine learning analyst. You could be go into something entirely different from biomedical science, develop applications, software tools, um, you know, for commercial use. It could be something entirely different from biomedical science. You could also be an ethicist, yeah. You could be a communication scientist, a medical, working in PR, you could work as an editor in journals, all right? So different fields, of course. You could also leave that and go into full-time medical science, right? So so many opportunities are there. Or you could be a patent attorney, going to doing a degree in law, master's degree in law. That makes you maybe a patent attorney, right? Yeah, that's usually applicable in Western countries. So these are so many opportunities available to scientists. I think this orientation is usually not given, especially for new students in 100 level, and they tend to be confused. Okay, after my microbiology degree, what am I going to do next? So they're confused, and that also um, reflects in their performances. Many of them do not end up getting good grades because they don't see the end from the beginning. So there's nothing to fight for, right? But so many opportunities are there if you can just be guided and be mentored towards achieving your objectives. So there are different transition pathways, just like I mentioned earlier. From being a scientist to being a lab director or professor, right? Or you could also ensure that your products should not just sit on the shelves. They go into commercial availability. They lead to product development. You could think about what you're doing now, what could be the end goal. This doesn't come, immediately. It could be a 10-year plan. That with my research, what can I achieve at the end of my research? What product can I develop? Right? What will be the outcome of what I'm doing? Am I just doing this to get a degree? Or am I just doing this to just get a publication and get promoted? Get a research grant, but you should also think about the end goal. Are you developing a product which even will make you richer? Because if you have a product that is available to use across the globe, then of course that it also brings money to you. So from branch to product development to commercialization, that should always be at the back of our minds when we are starting out our research, right? So you could work in the academia, like I said earlier. You could also work in the industry. So your decision is entirely up to you, right? But of course, in order to make an informed decision, then of course you need to really weigh the pros and cons of working in academia and industry. If you're going to be drifted by opportunities, or these were the opportunities, I have to go there. Yeah, that's also a decision to make. But if you want to a well-informed career decision, then you need to be able to weigh the pros and cons. In the academia, the pros, the advantages include that you're able to have get a degree that is largely recognized across the globe. And then you have more freedom of choice on this kind of subject you want to, or field you want to really go to, the kind of research project or research topic you really want to explore. That's a bit subjective because, of course, most times you tend to design your research in line with what the funders' expectations are. So, but again, you have so much time to be able to decide how you want it to be, to define your objectives, and then of course, the disadvantages could be that you may end up being overqualified for some job positions, especially if you want to now leave the academia to go into the industry. You have a PhD. PhD is not so much an advantage for you because, of course, when you work in the industry, they think more about experience and practical application of knowledge other than your degree. Then maybe you may be wasting your potential. This is kind of controversial because if you want to go into the academia, you spend most of your youth studying, you know, to BSc, spend years doing MSc, PhD. You can argue that, you know, you're spending most of your potential power and your youthful energy and, you know, resources, you know, just going to school. And then, of course, the end overall outcome of that, if you're thinking about the pecuniary or the financial benefits, then of course you think that that's not, you know, may not be commensurate with the commitment you've put into this. But if you're passionate about what you're doing, this is exactly what I want to do. I was not just drifted here. Then of course I want to believe that your passion will win in the end, and you will become successful and also be rich in a way. If you want to go into the industry, I think that has to be decided early. You tend to earn more money, especially if you go into pharmaceutical industry, or you want to start applying your new age, you know, techniques in AI, machine learning. You could work in Google, in Amazon, Facebook, or those big tech companies, right? And then of course you get to work on and solve real-time problems, real world problems, practical problems. And you know, when you go into the industry, maybe working in Pfizer, a vaccine company, you tend not to want to return to the academia because it's not going to be as rewarding as working in the industry. So the decision has to be made early enough. So I think I already talked about some of this. You know, the time commitments if you want to go into the academia, if you want to go to industry. And of course, if you're in the industry, publication may not be exactly required. Of course, it's desirable. It's desirable, but not a major requirement for advancement in the field. But you know, in the academia, that's a very important requirement. And then, you know, you need to publish, you need to write, attend conferences. So communication and mentorship really play a very important role here. And of course, it don't start sourcing for funding because most times the funds are there for you to work with. And if you're in the industry compared to where you're in the academia, where you have to spend days and nights, you know, writing grants, looking for money left, right, and center to do your research. So it's harder to work in academia compared to the industry. Speaking globally now, not specifically focused to the Nigerian context setting. So why does leadership matter here? Whether you're in academia or you're in the industry, you need leadership because if you want to grow in your field, you definitely will need to attain a status of leadership at some point. If you grow, and if you really want to grow, you need to now start learning about how to imbibe, cultivate the culture of leadership, right? Leadership drives innovation and impact, and the scientists, we must be able to communicate with other scientists, and of course communicate our research projects and findings. And of course, we should also be able to influence our environment, influence stakeholders, policymakers, decision makers, and of course, we should also be able to execute our projects, right? Implement our projects. So, as a leader, you should be able to supervise, coach, and teach. You should also be able to manage knowledge. You know, if you have mentees, trainees in your lab, you should be able to manage some kind of they're on fire. Let me put it that way. They're firebrand. So you should be able to work at their own pace. If you're too slow for them, they feel like, well, you know, especially the younger generation of very knowledgeable people. So if you cannot manage that kind of knowledge, then of course they find your laboratory not a very suitable environment to grow. You should be able to design your research methods. You should be able to manage people's attitudes and character, different qualities. That, if you have a mix of people from different foundations, their backgrounds coming into your lab, you should be able to manage people generally, and then manage their skills, and of course ensure that your project has a high impact, right? So that's why we need to be able to do that. So these are the four. This is not exhaustive, but there are four important skills you need to be able to acquire if you want to transition into leadership in science leadership. You should be able to communicate. You should be able to manage projects. You should have critical thinking, and of course you should have that sense of teamwork and collaboration, right? So we're going to be looking at each of them. So for you should be able to communicate not only with your lab members. You should also be able to translate complex science concepts into simple, clear messages, especially when you're having conversations with funders or policymakers, or even lay people, the entire consumers of your research, right? So you should be able to translate complex science into simple, clear messages. There's something called pitching, right? If you meet, for instance, you're flying, you meet someone at the airport or on the plane, you have to really, you only have 2 minutes to talk to them about your work. What are the key things you say, especially if you think these people or this person might be might be able to fund your research as an organization. For instance, let's say you meet Bill Gates and you need to talk about your very interesting research in maternal health, for instance. How are you going to communicate your research, even though it might be a bit technical person, but how you can communicate the importance of your research such that it's going to be interested. And I want to tell you that research funding is not only limited to writing grants, applying, competing with everyone, the open competition. Many of the funders fund scientists without even announcing or making any public advertisement for grants. So there's closed funding. There are so many closed funding opportunities. So if you're able to communicate your findings, you're able to communicate your research, then some of these opportunities will come to you, when, for instance, you go for a scientific meeting and during one of the side events, you have an opportunity to interact with one major science funder. And of course, he or she picks interest in your research based on how you've been able to effectively communicate what the importance of your work. So that's a very important quality that we have to really adopt. And of course, we should be able to really adapt our communication to different audiences. Are you really talking to a PI, a fellow PI like you? PI here means principal investigator, or you're talking to a potential funder or a policymaker, or you just talking generally to the public. For instance, if you go on Chinese TV, how are you going to really address your audience? You're not going to be using technical terms. You're going to break things down so that every person can understand the importance of your research and findings, right? And then there has to be that confidence when we're presenting our ideas at meetings and conferences, and of course, maybe at grand panels. So effective communication is a very, very important attribute, quality that we should have when we are navigating or transitioning towards leadership in science. So the other attribute is project management, right? We should be able to plan effectively. As a matter of fact, if you spend most of your time planning, you realize that implementation and delivery will be easier for you. You have effective planning. You already have everything mapped out. You plan your project, you plan your experiments, you've set your milestones, your deliverables, and you're able to balance the timelines and resources. And of course, you know that the other pieces will just fit in, the implementation and delivery will just, be a lot easier for you. So we should be able to plan effectively. We should be able to manage risk. And I tell you that there are a lot of risks associated with scientific research, especially in the biomedical field. The risk associated with the patients or participants, risk associated even with the entire experiment you carry out. Sometimes the entire assay fails. So it's very important to really be able to manage them. You have alternative plans, you have potential, you have this, envisaged or anticipated potential challenges ahead of um, uh, uh, the the initialization of your project, right? You're able to troubleshoot even if there are challenges, challenges from your projects, challenges in your mentees or students' projects. You're able to troubleshoot everything, they come to you, as your savior, and then of course you're able to really troubleshoot everything. Then you're able to monitor progress, document everything, and of course deliver results consistently, not just good ideas, but outcomes. You have to be able to deliver results, you have to be consistent. You can't afford to fail, so you're probably on the edge of most of the time, to just stay consistent, because if you run out of funds, then of course you won't be able to do the kind of research you want to do. And then of course, if you're interested in doing quality research, quality science, then you're not happy, you're not fulfilled, then you're not doing science, you can get promoted by using students' projects, to get small publications as well. But I'm talking, about in a general context now, but are you going to be fulfilled as a scientist? No. So you should be able to you should deliver results consistently, right? And of course, you should also be a critical thinker, whether you're in academia or in Israel, you should be able to analyze data objectively, and truthfully, right? You're not going to manipulate your data. And of course, you should be able to ask the right questions. You should be able to challenge assumptions, make informed decisions, right? You should be able to turn uncertainty into discovery, right? And that's your superpower as a scientist. So think deep, think your experiments through, your projects through, and of course, at every point in your analysis, you're able to make deductions, right? If this is the result, what does it mean? Go back to literature, find out the best interpretation of your results, because every result has its own inference or deduction that we have to make, right? So if our results are genuine, are real, then of course we should be able to make effective important interpretations from them, and that comes by being critical thinkers, right? Teamwork, yeah, this is very, very important, because you realize sometimes that if you're looking at the funding landscape, that's an entire topic on its own. The funding landscape is not so wide, because there are just limited funders or agencies interested in funding biomedical research. And it's even worse if you're doing research in Africa or in Nigeria. So what happens is that most EU funders or North American funders or maybe Asian funders, they want to work with you. They want to fund research, and they will fund their people. And what happens is that people can collaborate with you. So that way, some funding will trickle down to your lab if you're able to effectively collaborate with them. So you should be able to collaborate. From that spirit of collaboration, extend your tentacles such that if you don't get the funding yourself, you can be a co-PI. Maybe the funding is domiciled in one country in the West, right, or in the global North, generally, we could be a co-PI, right? And that comes with collaborating effectively. And you can only do that if also you have some substance in you, right? Maybe they know that you have knowledge or skills in maybe AI, bioinformatics, genetics, or who's doing important genetic research, maybe in sickle cell disease. They can Google, and then Google will give them your name, and then of course they can reach out to you, right? So that comes by also doing a lot of work on your own part. You should also within your own team now, in your lab, you should be able to resolve conflicts. There will always be conflicts because we have people from different backgrounds, homes coming into your lab. So you should be able to resolve conflict without having to appear one-sided. And of course, you should be fair and just in doing that. So you should be able to resolve conflict respectfully. You should also lead without being forceful, right? You shouldn't be seen as a megalomaniac. You shouldn't be seen as a tyrant or a despot. You should lead without force, right? Lead by influencing people, not by forcing them, not by micromanaging people. So you should be able to lead, and that comes by also being a good follower yourself. So you should be able to listen to other people. You should be able to be compassionate. Understand other people's points of view. So that's where you're able to lead effectively. So science is generally not a solo sport. So if you want to achieve breakthroughs, you have to work as a team. So in order to build all this technical depth, you need a strong scientific foundation. I mentioned earlier that to be a leader, it's not just by getting university promotions or promotions in your organization. No, you should also have some substance, right? There has to be that skill that you have that people want to learn from, right? Maybe you have knowledge of grant writing, or maybe you have knowledge of all these technical skills in bioinformatics or data science or machine learning. So people will come to you to learn from that. But if you're just you're just recruiting people who have these skills and you don't have the foundational knowledge, then of course they will take advantage of you because they'll know you're so dependent on them and you can't achieve anything without them. So they leave and then you're stranded. So you should have a strong scientific foundation. They should also inculcate the habit of continuous learn new things every day. There's a new tool in town. I need to know how to use it. There is a new technique in town. I need to know how to use it. There is a new way of writing grants. There's a new way of writing publications. Manuscripts. I need to really know how to do that. So that comes by getting recertified, upskilling, applying for courses. You can argue that so many of them are expensive, but there are also some free ones in Coursera. There are free ones in Coursera, and now we have AI tools that can really guide you. So many of them are also on so many of them are on YouTube, where people can learn from. And you should also learn to present at scientific meetings, present our research, no matter how little your research maybe you have that imposter syndrome. You feel, oh, this is not good enough. Maybe I shouldn't present. No, as long as you're able to present it well and you've been selected for presentation, please go ahead and present. Be visible so that people can see. I've heard so many stories of people going to scientific meetings, making presentations, communicating effectively, and then some founders in the audience just reached out to them for funding, and you supported them. They probably never got awarded any big competitive grant previously, but because they were able to communicate their research effectively and they were very confident, funders reached out to them and they got funded. And that was the foundation they laid their laboratory structure on. So we need to do this early. I don't know the demography of the audience, but of course we need to start early. We need to take initiatives. We need to volunteer for tasks, lead small projects in our teams. We need to propose solutions instead of waiting for instructions. We need also mentorship. This is also very important. We need mentors. You can randomly reach out to people. Well, sometimes send cold emails. I've read, I've been reading your work, or I've read your work. I've been wondering whatever it is, you want to coin it. And I'm interested in this too, particularly by this paper. And I was so excited and I would like to really, be your mentee. So maybe 8 out of 10 times you don't get a response, but of course maybe 2 people will respond out of 10, and that's probably going to be enough for us to advance in our careers. And then we also have to be mentorable. Right? We need to be able to learn from people ahead of us. We need to be able to ask questions. We need to act on feedback. I mentioned earlier we need to be able to build visibility, present lab meetings, be confident enough to answer questions during lab meetings, during conferences, ask questions after someone's presentation. Maybe you're not, you don't have the privilege to present your work, but after each presentation, they ask people for questions, be confident enough to ask questions then, that's part of building visibility. We need to be able to write summaries, share outcomes. LinkedIn is a very important tool, no matter how little it is. I always say that you don't let anyone define what you should post, or define how important your post should be. That is important to you and you want to share with everyone. So it's better for us to share outcomes, right? We need to be able to lead. Whatever stage you are, whatever level you are, there are people below you that you can mentor, be able to be able to lead, guide juniors. We need to be able to manage experiments, organize journal clubs. Those are very important skills to have. So I mentioned that networking and visibility, connect with people as much as possible. LinkedIn is there. Present at conferences, and we also need to publish our papers in reputable journals. So this is quite a lot, but it says a lot about building an effective professional portfolio, right? We should be able to showcase our work, especially if you do generate data science or work on data science projects. Showcase them on GitHub create a GitHub profile. Integrate your Google Scholar metrics in your LinkedIn profile or your email, stuff like that. Highlight your publications. I always say this. And then, of course, we need to be able to connect to real world problems. Tell a story. And in telling a story, you need to be able to provide the situation for your story, the task, the action you took, and the results, right? So we need to be able to use this STAR framework in telling our stories. We use visuals, right? Visuals are very appealing to the eyes. So, use visuals, screenshots, to highlight your metrics. Google Scholar is there, LinkedIn, GitHub. You can also generate your personal website. It's not expensive. You can generate your personal, I think you can generate free using, what's that? There is forgotten there are two or three websites that allows you to generate your personal website. So you can also create that if you don't have enough footprints on the internet. So when people Google your name, they're able to see some of the work you've done. So yeah, some other soft skills for leaders: negotiation. You should be able to negotiate without ruffling any feather. You should be able to, um, in collaborating, negotiate what is best for your laboratory, what is best probably for your team, your country, and you should also be able to make good quality decisions, right? You should be able to make decisions off the cuff. If you have to make decisions right now, you have to be able to think ahead and think, okay, what am I supposed to do? And you have to always get things right. I mean, if you don't get things right, it's okay. We're not always right all the time. So, but it's good to always make decisions that are very good for your laboratory. And conflict management, yes. If you're working in any setting, particularly in Nigeria, there will be conflicts. Conflicts could be within your lab. It could also be outside your lab. University conflicts, organizational conflicts, unnecessary bureaucracy, because you're working with people with different egos, right? You should be able to manage everything. It's a whole thesis on its own if you want to really do your research on that, but it takes being smart enough to really understand this. There are some you have to yield to some things. You have to stand your ground on some occasions, right? You have to pretend in some instances, right? Just know that the ultimate goal is for your work to be done and to be done in the right way. So we should be able to learn some of these skills as leaders. So we need to always look out for opportunities for growth, grants, fellowships. We shouldn't be tired of applying. We shouldn't listen to the second voice of saying that usually comes to tell us no, don't apply. No, I don't think you stand a chance. We shouldn't listen to that because if you don't apply, then you're 100% sure that you're not going to get it, right? So why do you want to be 100% sure that you're not going to get it? If you apply, then the probability could go from 0 to 100 for you. But if you don't apply, the probability is 0 to 1. Let's talk in technical terms now. If you don't apply, right, the probability that you don't get it is 1. Definitely.
Hello, sir. Can I just cut you short a bit? We
Have I
have
run
some
out of time?
questions. Yes, sir. Yes, we already sent a message.
All right, so I'll soon be done. Okay.
All right, thank you, sir.
Do you want me to take the questions now?
Yes, sir. You can take the questions. So anyone that has question for
Okay.
Dr. Oyebola
Anyway,
can drop
so I
them
only have 2 more slides.
while you answer.
All right, so this here. This is the action plan. So you need to identify your goals, build skills, find mentors, take leadership responsibilities. I have this Mentimeter poll, but it's okay. I already have questions in the chat, so I'll just not take this one. But you should be able to know where you are now, right? What do you want to be in 5 to 10 years? What skills, relationships do you need to get there? And you can write down 3 actions you will start this month. So network, and thank you so much for having me.
Thank you so much, sir, for taking us through this this interesting and impactful presentation. We have learned so much how to run our skills as leaders in the bioscience research space, how we ought to be strong communicators, build our teamwork, project management skills, and also critical thinking skills. Thank you so much, Dr. Oyebola, for this brilliant presentation. So anyone with question can drop it in the chat box. I would give an opportunity to someone to unmute and ask, and the rest can be dropped in the chat box while I check through and answer them. Thank you so much, sir, once again for this presentation. We really appreciate it.
Thank you. Thank you. Sorry for running out of time. I wasn't really conscious of the time, so but thank you for having me.
All right, so would also love to ask if you can share your presentation slides with us, sir.
Yes, I can forward my slides.
Thank you, sir. Thank you, sir. All right, thank you.
You
Any
probably have
questions
to
for
reach out to
Dr.
me.
All right, there are lots of
Maybe
shout outs to you in the chat box.
Oh yeah, thank you
Comment.
guys. Thank you.
Thank you. Any question? Thank you, sir. All right, in the absence of no question, if you still have a question, you can still drop them in the chat box while you attend to them, right? We will be moving on to the award and recognition part of our anniversary, and then I'll be handing over the mic to the co-founders right about now. Okay, we have one question from Wealth before we go ahead in our meeting. There is a question from Wealth, and I would like you to take it, sir. Just one question. How do we deliberately build scientific leadership capacity on the continent? What challenge do we need to surmount to do this? Right, you can answer that, sir.
Yes. All right, so how do we build scientific leadership capacity on the continent? So that we need to be able to first attract funding, because whatever we do requires funding. Whatever we do needs funding. To build capacity, we need to recruit people, students from at different levels, trainings at different levels. So we need funding. So for us to be able to, whatever we need to do to attract funding, we have to do it. We have to build a reputation as scientists. That's a very important factor in deciding whether or not to be funded. We have to acquire important skills, right, related to our field. Of course, important grant management and grant writing skills. And of course, we have to also work in an environment that allows for us to be successful in attracting grants and also in managing the grants, right? So these are very important things. You can argue that all these three conditions may be difficult to achieve, especially on the continent. But there are some institutions that allow growth. There's some institutions, even on the continent, that allow growth, environments that will enhance your competitiveness for research grants. So we need funding, first of all, to be able to build capacity. And then there'll be a lot of challenges. Yes, we have quite a number of them. Funding is one of the challenges. There's limited funding. Okay.
Sir, right. Could you please stop sharing your screen? All right, sir. Thank you, sir. Yeah, you can continue with the answer, sir.
Oh, okay. I thought my time was up. How much time do I have, please? I really don't want to overshoot my time.
You can round up in a minute, sir. A minute, sir.
Okay.
Yes, just yes.
So yes, funding is very important. Funding is a very important limitation because we don't have enough of that. And of course, the environment, apart from the infrastructural limitation, the people who in leadership are not so well exposed to encouraging research and capacity development, especially in most institutions. So we have people in leadership that are not so well exposed or in tune with leadership and research leadership and capacity development. So those are very two critical factors. We don't have any challenge with human resources. We have very brilliant intelligent scientists that can learn anything and can develop anything. So those are the two important, from my own point of view, funding and research environment. Those are very key challenges we have at this point.
All right. Thank you, sir. Thank you, sir, for the response. If you have any subsequent questions for Dr. Oyebola, you can drop it in the chat box while he attends to it. So right about now, we will be going to the award and recognition by the co-founder. All right. Handing over the mic to them. They can unmute and take over. Thank you. Hi, Wealth.
Yeah, thank you
Martin.
so much, Gloria. Can you hear me?
Yes, I can hear you clearly.
Okay, thanks. Okay, so I'll be sharing my screen now. So can everyone see my screen?
Yes, we can see it. We can see it. Yes, we can.
Okay. There's a poll that is up. I don't know if I should.
Okay,
Okay.
yes, there is a poll to get our feedback for the anniversary. So we can just quickly attend to it, and then it will not take our time just to get our feedback concerning the two sessions. Thank you. The 2-day
Okay.
sessions. Thank you.
Yeah, so thank you so much, Dr. Oyebola, again for the very detailed and in-depth presentation. Thanks for sharing your perspectives with us. And to all the amazing guests we've had today, the poster presenters, Dr. Shakir, Mr. Raphael, Aminu, Adegboyega, and Paul, thanks so much for sharing your work and sharing your perspectives with us. So we are already wrapping up the anniversary event, and we are really excited that our community has come a long way. It's our 5th year, and we are also very excited for what is to come for us as individuals and also as a community. So we thought it will be great to take this landmark event to just appreciate some of our members who have demonstrated exceptional leadership, service, and volunteering capacity. So we typically recognize all of our volunteers and team leads during anniversaries, but this time we just want to kind of just recognize some of our volunteers and leaders and members who have just kind of gone over and beyond in their commitment to the community, and to just encourage them that we are seeing their good work and that they should keep the flag flying, as it were. So I will not be doing this alone. I will be doing this alongside Dolapo. I hope he's still with us. So maybe Dolapo can pop up and say hi.
Yes, thank you very much, Wealth. Very much, yeah. And yeah, big thanks to all our speakers, like what I've said. Really, really enriching conversation tonight. Really appreciate that. To the poster presenter, thank you very much. And yes, like Wes said, we're just going to be looking at recognizing some of our members. Of course, we've had amazing, of course, we have amazing members in Biospherea. But we're just going to be recognizing, some persons tonight, and that will be well myself, giving out this award. So yeah, thank you, Walt.
Yeah, sure. So we will be starting with our founding members. So these are people that have been in this committee for 5 years. 5 years is such a very long time, and not just been in the committee for 5 years, but they've made very, very remarkable impact within that period. So we'll be recognizing 7 of our founding members, and we'll be appreciating them for their impact in the community and their activity, how active and committed they've been in the community. So these are people that even when we call their names, you would know, okay, these people they've done so much. They provided mentorship to some of us, they provided guidance. These are people that we can slide into their DM at any time of the day, and they respond to, requests to take up new responsibilities and roles, and they've just been so instrumental in us building the community into what it is today. So we'll be
So we did something quite interesting. So we have like a general message from the community, but we also reached out to individuals that have worked with these people or be mentored by them, be supported by them, their friends, to just say, oh, what can you say about this person? To just add a layer of, spice or beauty to what we'll be doing this evening. So we'll be starting off with Abdulafiz Akinloye. And so you can just read what's on the slide. I'll also be reading it out because I think there's something beautiful about hearing people say certain things about you. So we are saying a very big thank you to Abdulafiz Akinloye, one of our very amazing founding members. So Abdulafiz has been an enviable example across several laudable metrics, and you've excelled at showing all of us what it means to be fully, actively, and purposefully present. So thank you for your mentorship. Thank you for your insights. Thank you for your guidance to us as a community, from the founders to our volunteers to every member. And then Shakir has a lot to say about you, but we just took this section of what Shakir Atoyebi wrote about you. So he said he has always admired Abdulhafiz's hard work and kindness. They've known each other for several years, well over 10 years, I think up to 16 years. And so that 15 plus people, including myself, that's Shakir, have joined the community through Abdulafiz, speaks volumes about his dedication to the group and reputation among his colleagues. Through both challenging and good times, his commitment to group activities has remained strong. He always gets the job done. And we all know that even though Abdulafiz is in Australia, it's probably like very early in the morning in Australia. Abdulafiz is one of our most punctual members to every meeting. So this is so much for him, and we do not take it for granted that he's bringing this level of commitment to our operations as a community. So thank you so much, Abdulafiz, and we look forward to building with you for many more years to come. Thank you. And next we'll be appreciating Chinaza. Chinaza is also a founding member. She's also the lead of the Legacies team, which is our partnerships team. So Chinaza is a team. That's something we are all familiar with in the community. So that may sound like a contradiction. How would one person be a team? But there seems to be no better way to capture our immense labors and contributions to our community. So Chinaza, you wear so many hats and do so with much poise. Thank you for showing us that it's possible to wear so many hats, to juggle so many things, and to be excellent at all of them. So we reached out to Ayomide Faloye to say something about you. And this is just a section of what he wrote. So he says, Chinaza is one of the most efficient people I've worked with. She has a unique way of ensuring tasks are completed with exceptional diligence. And she always finds a way to bring everyone along. So Chinaza, well done. And thank you so much from myself, the founders, and the entire community. Thank you so much. So next we have Nasiruddin Abdulrashid, a founding member and a very active volunteer. So Nasiruddin, your consistency, your warmth, and your charm endear you to us in so many ways. You bring such unique angles to conversations that it has become impossible to not notice your incredible passion for the advancement of science in Africa, one that fearlessly shines through your commitment. We are proud to be building alongside you. Thank you. Thank you so much. And Ruth Totti, who you've worked with for several years, has this to say about you. I don't know a more committed member with flawless attendance records and consistency. Nasiruddin is not only an active member and volunteer, but also one of our biggest advocates. I think everybody can, say a very big and resounding yes to that Nasiruddin is both active as a volunteer and a very passionate advocate for our community and what it stands for. Next, we have Esther following show, and I think we should be giving this award to Esther alongside our very young scientist daughter of us, who would be a member of Biospherea, who is already an associate member of Biospherea, but hopefully will be a full member in several years to come. So Esther, you've strengthened engagement across multiple teams. You've worked in chaperones, you've worked in camps, you've functioned in anniversary planning committees and a host of other committees. You've co-organized projects and provided mentorship. All these and more you've done without losing that bold spark of joy with which you fill every space. So thank you for sticking with us and staying true to our shared values all these years. We hope most of you are still on the call, but if you're not, we'll encourage you to watch this video and, also just read some of these things that your friends and colleagues and teammates have to say about you. So we reached out to Olamide Oduwole to say something about Esther, and Olamide did say that Esther was one of the first persons she worked with in a team, presentation team, shortly after she joined the community, and she described Esther as a gem who brings dedication, passion, and a joyful energy to the team. Everybody knows Esther is that very bubbling person. She is also always willing to lend a helping hand and offer great advice with a warm approach. So that's from Olamide, who has been your team member and colleague while you were still a chaperones volunteer. And then we have Victor Olagunju, a founding member and volunteer who's also been very instrumental in planning this year's anniversary. So Victory reminds us that strength is strength, whether or not it is fierce and firm or quiet and unassuming. So thank you for not only seeking, but also embracing opportunities to build and lead with us, and for doing so with premeditated preparedness. So Victory Titilayo has this to say about you. So Victory is a rare gem. So this is just a brief section of what Titilayo wrote. She wrote like multiple paragraphs. Victory is a rare gem and has shown himself to be both helpful and valuable despite busy schedules. And then Esther following show has this to say about Victory, that you're an excellent communicator and a great team player. So thank you so much, Victory, for your support. Next, we have Ruth Doty, our founding member and the Chaperones 001, our community manager lead. It takes a lot to perform and excel at your role, Ruth, but you've made it seem so easy, so enjoyable. Your devotion to duty, not only as a supportive leader, but also a bold advocate, continue to inspire us. We've seen your impact time and again, and we know there's even much more to come. Thank you so much, Ruth, for your commitment. So we reached out to Adede, who is one of our moderators for today, to say something about Ruth. And here's what she has to say about you, Ruth. She said, Ruth is kind, warm, and caring, and her leadership is inclusive. When I joined the team, she instantly made me feel like we could walk through everything together. And I think that's how Ruth makes every one of us feel in the community as our community manager. Next, we have Olaiton Owoyemi, a very amazing member and volunteer. Olaiton has volunteered in anniversary planning, volunteered with social media, now volunteering with legacies, and he has done so many other stuff behind the scene that some of us will not be aware of. But when we think of strategy, when we think of structure, and when we think of execution as a community, you readily come to mind. Little wonder you played a major role during our transition into Biospherea in 2022 and 2023. Thank you for consistently delivering on excellence all these years, and for always reminding us that we can always have bigger, better, and bolder dreams. So for those of us who have served as leads and volunteers, you know that when we have these stakeholder meetings, Olaito is always talking about all the things that we can do as a community. And he has been very consistent. If those who are like founding members, like right from 2021, Olaiton has been dishing out so many big stuff that the community could venture into. And we'll definitely venture into all of that in a few years to come. But we've just been so inspired by your perspectives and contributions as a member, a founding member and a volunteer. So Chinaza, who you've also worked with for a long time, for a long time, but more recently as the legacies lead, has this to say about you. He said Olaiton is very reliable. She actually said reliable like 3 times in the short paragraph she shared with us. So Olaiton is very, 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 very reliable. Working with him, you come to appreciate the kind of person that always gives his best. So thank you so much, Olaiton, for always and consistently giving your best. So that's all I'm going to take for our founding members. So I think Dolapo would come up now to appreciate the other individuals who will be recognizing this night.
Right. Thank you so much, Wealth. So the next category will be the volunteering award. So basically, these individuals, we've looked at impact, the duration of volunteering. We've also looked at the ability to, initiate, take initiative, and also their consistency. And these were the criteria we looked at. And the first person on the list, I'll be, talking about the award tonight, is no other person but the person who led the BBRP project, this year. Very, very amazing individual. His friend has described him as a phenomenal and a brilliant scientist, and someone who is wanting to always make impact wherever I find himself. So thank you so much, Adegboyega, for your incredible contribution to our community. And then for the initiative of the Biospherea Budding Researcher Program, this program, impacted over 10 members. They were able to develop their research skills and research competencies. So thank you for simultaneously setting and raising the bar. And yeah, we reached out to Ololade Gbadebo. He said that Adebayo is a brilliant and phenomenal scientist who make impacts wherever I find himself. So thank you, Adegboyega, for your contribution. The next individual we are going to be recognizing for the volunteering is someone who is a steady, a long-time volunteer. You know, he has been very vocal when it comes to advocating for undergraduates. You know, I can remember those days, you know, when we have presentation and Esther would be like, oh, we need to, you know, tailor our presentation so that the undergraduate can also have, you know, chance to learn. So she's been so vocal when it comes to, you know, advocating for undergraduates when she was an undergraduate herself. We really appreciate your commitment, your growth readiness, and then, you know, you stand out in so many ways. We're glad to be a part of your journey towards becoming. Thank you so much, Esther Adefoju. And yeah, we did reach out to Ruth, who said that you are an advocate volunteer and you have grown to become one of our best moderators. The way she embraces the community's values and vision is admirable. Thank you so much, Esther, for your incredible contribution to our community. The next individual who we are going to be recognizing under the category of volunteering is someone who has always helped us with generating insights, you know, in terms of detailed survey reports. And then we take this insights to make informed decision. Is no other person than Ibrahim Adebayo Azan. Very fantastic individual. You know, you're always ever willing to take up new challenges, but you rarely stop there. This is someone who goes beyond and above, trying to set, you know, nothing short of excellence. Thank you, Brian, for all of your hard work during these past years. We really, really do appreciate your contribution. And Abdullah Fiz has this to say about you. He said there's always something new to learn from him. This is also matched with his humility and willingness to learn. Admire Ibrahim a lot. And yes, thank you so much, Ibrahim. We really appreciate it. Now, the next person is, you know, is a camp lead. He has volunteered as an advisory committee member. She's also, you know, very, very strategic with insight and advice. She's an efficient team and activity coordinator, and she ceases not to, you know, take initiative. It's no other person than Charity Okola. She was a former lead for CAMS. Apology for that. Not a current lead. She was a former lead. But yes, she's demonstrated exceptional leadership during that time as a lead for CAMS. And Charity, thank you for being a firm support pillar, an executor, and an inspiring teammate. The depth of expertise and breadth of experiences have helped us to integrate structure and stability into our operations. And Alimat has this to say about you, the current camps lead. He says, working with Charity puts you on your toe in a good way. She always seems to have a lot of energy, which makes you to want to have a similar level of devotion. And that's a fantastic one from Alimat. Thank you, Charity, for your leadership. We really appreciate it. So the last but not the least on the volunteering certificates is another fantastic member. Is a camp member and is a long-term volunteer, over 4 years in camps now. You know, he has the initial stint in synthesis, and he was the one who proposed the volunteer spotlight for social media. No other person. Please appreciate Sheriff Udin Ahmed. Is a volunteer of camps. And again, another fantastic individual. So we've watched your growth over the past 4 years. Admired your tenacious consistency, marveled at your devotion to excellence, but even more at your selflessness. Thank you for saying truth. And yeah, Charity said, Sheriff Dean is deeply committed to the growth and well-being of the community. His consistency, thoughtful contribution, and willingness to support makes him invaluable. So yeah, thank you, Sheriff Dean, for your amazing contribution to our community. And so, yeah, that would be it for the certificate of volunteering recognition. And now we go to the next one, which will be the leadership category. And for this, we've looked at
Hi everyone, can someone hear me?
Okay, can you hear me now?
All right, yes, we can hear you, Dolapo. You
Yes,
can go on.
apologies. Yeah, I've just got cut out from my Zoom. Yeah, but I'm back now. Yes, so that would be it for the volunteering. So the next would be the leadership category. And for this, we've pretty much looked into criteria such as so basically, we looked at mentorship, impact, ability to take initiative. We've also looked at their coordination and management skills. We looked at communication, duration, and the efficiency. And the first person on this category is someone who has demonstrated exceptional leadership. Yeah, was demonstrated exceptional leadership. She's the CAMS lead, and when you see all of our communications, LinkedIn, this is the brain behind those activity. Very fantastic individual, and that goes to Alimat Chizomatunda, and very very incredible leader. Your leadership has not only provided nurturing growth environment for CAMS, but also demonstrated a nigger readiness to take up new challenges. Give your best and outdo yourself each time. Thank you so much. And Brian Obed, as this to say, Alinat has been inspiring. She's someone who everyone loves to learn from. She has pushed all of us and the counts team to be better. And someone also has this to say. A member says she's friendly to work with, and that's really really fantastic to hear. Alimat is an incredible leader. Thank you so much for all you do for Biospherea. So the next for the leadership is no other person, but this individual, as single-handedly, he has led his Jonah Club with so much passion and creativity. And when I look at the Jonah Club, the drug discovery, and I looked at the activity, this individual has carried everyone along. He's demonstrated enormous mentorship, supported people, communicated effectively, carried everyone along. And is no other person than the lead of the drug discovery and pharmacology journal club, Dr. Shakir Atoyebi. Really, really amazing this, past year. Thank you so much for your leadership, particularly for the journal club. But not just that, we really appreciate your insight, your support, in moving Biospherea forward. Your skillful commitment to stretching, supporting, and shaping the next generation easily position you among the many giants we should have become stepping stones towards realizing our dearest aspiration. Thank you so much. We reached out to Nasruddin Abdul Rashid, and he said, Shakir embodies servant leadership characterized by empathy, mentorship, and commendable values. He is a valuable asset to our community. So thank you so much, Dr. Shakir. We do appreciate your amazing leadership for our community. And that will be it for the certificate of recognition. Thank you, everyone.
Thank you so much for we the amazing members, amazing leaders have been appreciated, and then let's give a round of applause for them again. Let's just show our excitement for those people that have contributed to the growth of Biospherea. All right, before we go on to the closing remark, I would like to do a shout out to the moderators so far for the 2 days, and I want us to show our excitement and appreciate them for how they have moderated well. I want to appreciate Ololade and Olamide yesterday. They moderated and it was superb. And I also want to do a shout out to my co-host today, Adede. Let's just appreciate that, and also to myself, my small self. All right. So to close this anniversary, I would like to call on Augusta to give the closing remark for the fifth year anniversary of Biospherea. I would be handing the mic to Augusta. Augusta, you can unmute and take the closing remark. Thank you.
Hi everyone, just to confirm that you can hear me.
Yes, we can hear you clearly.
Okay, great. That was so exciting. Thank you so much to the co-founders. That was really, really exciting. You got us very excited, and congratulations to all the awardees. You guys are really, really doing great. So I've been asked to give a closing remark and acknowledgement. In no particular order, I would like to acknowledge the anniversary committee. Thank you so much for all the work that you put into making the past 2 nights go really well. To the programs team, thank you so much. I'd like to acknowledge you all. To the Biospherea volunteering teams for their cross-functional team collaboration. We worked with so many people. Thank you so much. I acknowledge you for all your hard work. To the founders who have surprised us this evening, Martin, Dolapo, and Wealth, thank you for all that you do. Without your visionary, without your dream, we will not be gathered here today. So we acknowledge you and we thank you for being the founders of this space for us to share. To the speakers, thank you so much for taking out the time to join us yesterday and today. Thank you for your presentations, and thank you for also sharing your wealth of knowledge with us. To the poster presenters, thank you for sharing your research and for also making submissions to share your work with the Biospherea team. To the community of amazing, amazing members, being in Biospherea all these years, I've met so many amazing people. Some of you were awarded tonight. Congratulations again. Thank you to every community member and to also participants that have joined us from far and wide. Thank you so much. We acknowledge you and we hope to see you next year on a bigger and better scale. To Biospherea, to all the community members, to our founders, we say a big thank you for being here. And with that, I would close the event.
Thank you, Augusta. Right. We're closing to meet next year. Good night, everyone.